Hello everybody. First of all, thank you to the organizers and thank you for also staying till late. Today I'll be talking about bacterial type 4 secretion system. So I'll take you back again from the mammalian system or eukaryotic system to the microbial world. So type 4 secretion systems are bacterial molecular machines. Bacteria has about about 9 different types of secretion systems to transfer macromolecules through the cellular envelope. These are from type 1 to type 9 secretion systems. But arguably type 4 secretion system is the most versatile and the most elaborate. Why I say so is because bacteria use type 4 secretion system to transfer small molecules, peptidoglycans, nuclear protein complexes, and a vast array of different toxins or effector proteins. Today I'll be talking about this particular bacteria, Legionella pneumophila. That actually utilize type 4 secretion system to transfer 300 different types of effector proteins. So it's a single molecular machine which is transferring 300 different types of effectors. These molecular machines are present in gram-negative bacteria, gram-positive bacteria, and even in archaea. So they're quite well conserved and they do a lot of different things. So in terms of function, bacterial type 4 secretion system has been classified into 3 different types. So we know about bacterial conjugation process. So bacteria exchange DNA between 2 bacteria, and this is done by the bacterial type 4 secretion system. This is another process which is called the competence system, where bacteria can take DNA from environment into the cell or release DNA from the cell to the environment. This is also done by the bacterial type 4 secretion system. And finally, bacteria also use type 4 secretion system to translocate vast different types of effector molecules into eukaryotic cells. For example, Helicobacter pylori utilize type 4 secretion system to inject CAG-A, which is an oncogenic protein, into eukaryotic cells. Similarly, there are other bacteria, for example, Bordetella, Brucella, Bartonella, and the list is quite long. And all these bacteria utilize type 4 secretion system to inject effector proteins into host cell. There are many different types of disease that also happen because of type 4 secretion system. The list somehow got cropped. But 2 of the things I wanted to really highlight is that stomach cancer or the peptic ulcer, that's generally because of the Helicobacter pylori that injects CAG-A protein into eukaryotic cell, and that's why we get this. Another thing we are now hearing a lot is antibiotic resistance, and that is also another reason to study type 4 secretion system, because the spread of antibiotic resistance is primarily occurring because of the type 4 secretion system. So that's in terms of function, there are 3 different types. If we just look at sequence and genetic organization, there are 2 different types of type 4 secretion system in terms of the number of components and the sequence. Type 4A secretion system and type 4B secretion system. So type 4A secretion system has 12 components. These are VRB 1 to 11 and VRD4. And type 4B secretion system, in contrast, has 27 components. So it's almost like twice as big. Both the systems are driven by 3 ATPases, which are colored in blue. And although both of them are type 4 secretion system, the only real homology between the 2 systems comes in this protein, which is called VRB10 in type 4A secretion system and .G in type 4B secretion system. So this has 12 components, this has 27 components, but really one protein is shared between the 2 systems. For type 4A secretion system, we know structurally quite a bit. For example, we know how exactly the outer membrane complex looks like. This is about more than a megadalton complex composed of 3 proteins, VRB7, 9, and 10. We have also a low resolution reconstruction of the core part of the complex, and the latest breakthrough came from Gabriel Waxman's lab when they solved the structure of the type 4A secretion system. This has 8 out of the 12 components. This is a negative strain reconstruction. This is, but just by looking at it, you can see that it's quite different from any other secretion systems. The outer membrane would be somewhere here, the inner membrane is somewhere here, and then these are 2 ATPase barrels associated with the inner membrane. And then this is the core part of the complex, this is outer membrane complex, and this is inner membrane complex. Both these structures are at much lower resolution. It's about 2.5 nanometer resolution. But still we know quite a bit about the type 4A secretion system. For type 4B secretion system, we actually know very little. The best we know is that these cells, Legionella pneumophila cells, they have type 4B secretion system, and if you osmotically shock these cells, then everything comes out of the cell. And then if you look at these cells using negative stain, you see these ring-shaped structures on the cell surface. And if you delete those, delete the type 4B secretion system from these cells, then you don't see those rings. So that just means that these are type 4B secretion systems. But we really don't know anything other than that in terms of architecture of this type 4B secretion system. And as I said, that they have like about 27 components compared to these, which has 12 components. So what we wanted to do is that we wanted to get the structure of type 4 and type 4B, both type of secretion system inside the cells, and understand whether type 4A and type 4B secretion system are architecturally related, because they are both type 4 secretion system, but the number of components and compositions are quite different. And we also want to know the assembly pathway and architecture of the type 4B secretion system. And our method of choice is electron cryotomography. Why electron cryotomography? Because there is a certain advantage of this. First of all is that we can look at the protein complex in this near native state and in the original context. So basically, there is no artifact of purifying a complex, because when you try to purify some complex in detergent, this complex might fall apart, it might get squeezed. So there is no such artifacts. And there is no staining related artifact or dehydration related artifact. So, and you can also look everything in the near native context, so that's the best part of it. So what do we exactly do? I don't think we really need to, ideally need to give much introduction to this audience, but still, so here is the grid. We have the sample in, and basically we just take 2D projection images of this grid at multiple different angles. And once you have taken all these different projection images, we just put them back together using back projection to generate a 3D volume, which is called a tomograph. So this is just a schematic of the same thing. Now I show this for a reason, is because suppose we are collecting data here 60 degree plus minus. Because of technical limitations, we cannot collect data here. So we cannot really collect data plus minus 90 degree. So what happens is that since we do not have information in this part of the tomogram, when you back project these to generate the 3D volume at the end, it's not a perfect reconstruction. There's still some information missing. And this is what is called the missing wedge. So if you could collect a 85 degree plus minus 2D projection images of an object, then it would almost become a sphere after reconstruction. But as you go lower in angles, for example, this is 60 degree plus minus, then you miss out some information here and here. You go further, like 45 degree plus minus tilting angles, you lose a lot more information here. And this is called missing wedge. I just want to remember that there is a missing wedge and problem, and then that can lead to a washing out of information, because this will become little important for the later part of my talk. So anyway, we collected some tomograms of Legionella pneumophila cells, and this is bacterial outer membrane, this is inner membrane, and we start seeing some electron dense cone shaped structures that are extending from inner membrane all the way to the outer membrane. So you can clearly see these. And we know that these cells produce type 4B secretion system, so that kind of suggested that we can really see type 4B secretion system in these cells. This is just a tomographic slice of this tomogram, and we can really see electron dense object going from inner membrane all the way to the outer membrane. This is one of the top views of this complex, and there are like 2 concentric rings. So if we just delete the type 4 secretion system from these cells, then we don't see these electron dense objects. So that's suggested that these are really type 4B secretion system in Legionella. So these molecular machines generally assemble at the cell pole. So this is a fluorescence image, and you can see that they're always at the cell pole. But sometimes we also saw that they also assemble at the mid cell. They are generally polarly localized, so why would they assemble at the mid cell? So it became very apparent when we just take all these different images and do some analysis on them, that when you just look at a normal cell length, then they are at the cell poles, but as the cell length increases, then something appears at the middle part of the cell. And then eventually the cell divides into 2, and then these are 2 daughter cells. Again, they have fluorescent puncture at the cell poles. So, so it is very clear that something was appearing at the mid cell when the cell was trying to divide. And when you take tomograms of the cells near the division plane, we surely saw some type 4 secretion system near the division plane. So it appears that while the cells are trying to divide, by that time already these molecular machines are getting targeted to the cell poles. Anyway, just going back to these particles, so if you look at one of the particles and then zoom in into the particle, then we can start seeing some very distinct features. For example, there is an electron dense layer just below the outer membrane. There is another layer in the periplasm, and then there are some more densities here and here, some densities here too, but if you're not looking at these tomograms every day, it's difficult to really appreciate these densities. But I think what is very apparent is that there are two densities. One is just below the outer membrane, and one is at the periplasm. And it looks like almost the Wi-Fi signal, so we call these Wi-Fi particles. And so this is just by looking at individual particles, we can already start seeing a lot of features. So what you can do is that you can take many such particles and then do an average of that, which is called a subtomogram average. So this is a subtomogram average of the type 4B secretion system in Legionella. This complex is quite flexible. So when we align these particles only on the outer membrane, then we cannot see any density in the inner membrane part. And similarly, if we align focused on the inner membrane part, we do not see any density in the outer membrane part. So we basically align both the parts separately and put together to make a composite model of the complex. It's just an estimation, rough estimation of how flexible these complexes are. So if we just align all the outer membrane complexes and then put a single dot for that, these are all aligned, then the inner membrane complex is, this is the distribution of the inner membrane complex. So this just shows that how much flexibility is there between the outer membrane and the cytoplasmic complex. And similar thing was also observed for the type 4A secretion system structure that I showed a few slides ago. So this outer membrane part and the cytoplasmic complex, they are quite flexible. So it seems like an intrinsic property of the type 4 secretion system. Anyway, from the subtomogram average, we actually identified many distinct densities. For example, we call this density as alpha, this one as beta. There's a hat-like structure. Of course, these cells are grown in broth culture, so they are not in their active state. So this mouth is closed here, so we call this hat structure. And then the outer membrane complex is connected to the periplasmic complex to this structure, which we call stem. And the whole outer membrane and periplasmic complex is connected to the inner membrane through this hazy structure, which we'll call the stalk. And there's some lighter density here, which we call wings. In the cytoplasm, we just see there are 4 lines. We just call them cytoplasmic rods. This is a resolution estimation of this complex, which is generally between 2 to 4 nanometer resolution. So next step was that we wanted to really know where exactly the core part of the complex is. So previously there are some groups which identified 5 different proteins that forms the core part of the complex. So what we did, we deleted everything else than those just 5 proteins. And those 5 proteins actually assemble inside Legionella cells. So this is almost like in situ reconstitution. And they form the core part of the complex. We can get a subtomogram average of the complex. Here I'm just showing that the 5 proteins expressed separately, they are actually expressing to the level of wild type. And what we can do is that we can really take this core part of the complex and position it in the wild type full length complex. And this shows us where exactly the core part of the complex is. And this is the difference map between the wild type and the core complex. So the black part here is the core complex, and the yellow densities here are because of the other proteins. So this is, as I say, this is the structure of the type 4A secretion system. This is structure of the type 4B secretion system. This has 12 components. This has 27 components. But architecturally, it seems like they're quite similar, because they have like this outer membrane complex, then there is a periplasmic complex, and there is a stalk that connects these two to the cytoplasmic complex. And then at the cytoplasmic side, there are like these 2 barrels. So we take a cross section of a barrel, that just means 2 lines. So there are 2 different barrels, so this appears as 4 lines. So our interpretation was that in our subtomogram average, when you see 4 lines, that just means that there are 2 barrels. So architecturally, the type 4A and the type 4B secretion system are quite similar. However, if you put them in scale, then the type 4B secretion system is much bigger than the type 4A secretion system. It's almost like twice as big. So as I said, that the densities in the outer membrane resolve very well, so we can really see distinct features in the outer membrane part, but we didn't really see much in the cytoplasmic part of the complex. So here we just saw like there are 4 different lines, and as I said that these are all because of the primarily because of the 3 ATPases, the blue colored ones. And we have confirmed that also further when you delete all the 3 ATPases, then these lines go. So we know that these are because of the ATPases. So we thought that since this is not really the active state of the complex, so that's why maybe the ATPases are not very stable, and that's why we cannot really resolve this very well. This is probably dynamic, flexible, and in the inactive state. So we are trying to explore many different other bacteria and type 4 secretion systems in those bacteria, trying to see if we can find an active state of the complex where it will be engaged with interacting with the host cells. So in that case, the cytoplasmic complex would be maybe more stable, and then we can probably get more information out of that. So we tried many different types of bacteria, but what I'll show is what we learned from this particular bacteria, which is Helicobacter pylori. As I said, that Helicobacter pylori also inject CAG-A protein into the eukaryotic cell using type 4A secretion system. So we thought that if we can image this interface when the bacteria is engaging with eukaryotic host cell, then maybe the complex will be in its active state, and then we'll be able to get more information about the cytoplasmic complex. And maybe the complex will also resolve much higher resolution. So what we did is we just grew gastric epithelial cells on EM grids and infected these cells with Helicobacter pylori cells. So this is the edge of one gastric epithelial cell, and then this is a Helicobacter pylori cell which is interacting here. So we collected tomogram of many such interfaces where the bacteria was really interacting with eukaryotic cell. This is one such section from one such tomogram. This is a tomographic slice. So very interestingly, what we saw is that whenever we co-cultured Helicobacter pylori cells with gastric epithelial cells, they always produce these long tubes. These are membranous tubes, and if you just grow them separately, the bacteria, then we'd never see these tubes. These tubes have they have some port-like structures at different places, and these are quite open. These are membranous, of course. And what was interesting is that we many times saw type 4 secretion system immediately next to it. But the fun thing is that, or interesting thing is that we never saw type 4 secretion system just below these tubes. Although if we delete type 4 secretion system components, then either the tubes are gone or their lengths vary with different mutants. So they're definitely related to the type 4 secretion system, but we don't really know how exactly they are related and whether these are also used for translocation of effectors. So we also got a subtomogram average of the Helicobacter pylori type 4 secretion system. And if you just compare that to Legionella type 4 secretion system, then it has also this similar kind of density, alpha, beta, gamma, and then stalk, and then a cytoplasmic complex. So again, Helicobacter pylori and Legionella also seem to be, they also have a similar kind of architecture. We also saw many top views of this complex, and then we could find that these are 14-fold symmetric. Now, what about the cytoplasmic part of the complex? Because that was our main aim, that we wanted to get it high resolution, and we wanted to learn how exactly the organization of the type 4 cytoplasmic complex is inside cells. So if you look at Legionella cells, there are these 4 lines. But when we look at the Helicobacter pylori type 4 secretion system, we see like a cup-like structure in the middle, and then there are 2 lines at the both sides. So it doesn't seem like both the cases it is same. It's quite different. And I have to mention here that here the number of particles is almost 10 times less, but still it is almost at resolved at very similar resolution, or maybe this part is resolved at much better resolution. So possible that when they are trying to engage with the host cell, maybe the complex is more stable. So a subtomogram average is of course it's a 3D volume, so you can look at multiple different layers. So this is the central slice. So if you look at top and the bottom slice, at different slices actually it looks like 4 layers. So top and the bottom slice, it looks like 4 lines, but in the central slice it looks like 2 lines, and then there is a cup-like structure in the middle. And this is the end-on view of this complex. By end-on view, what I mean is that you are looking at the complex from the cytoplasmic side. And here you can see that there like looks like two lines. So they're like this one pair of line and there is another pair of line, and that's where the concept of missing wedge comes. So since inside cells, when we are trying to image these molecular machines, we are not getting the full information. So when you are looking at a sphere, and after 3D reconstruction, if you have a say 60-degree plus minus data, then of course you're missing out quite a bit information along these particular axes. And our interpretation of this data is that it's not just two lines, it's like one full circle here, another circle here, another circle here, another circle here. And because of the missing wedge, you're just washing out the information along the axis. So we basically wanted to know how exactly this whole thing is organized inside cells. So for that, what we did is we generated many different volumes and then collected tomograms, basically simulated tomograms of these different volumes at giving a 60-degree missing wedge, and then look for the different signal that we get from all these different